Okay? So as the blood carries your glucose to the cells of the body where it is transported across cell membrane by carrier molecules. So you have a transporter there. So the above process is facilitated by insulin mainly in adipose tissue and muscle tissue, which means that the target tissue or target organs of insulin is the adipose tissue and your muscle tissue. Okay? Now, transport of glucose is not facilitated by insulin in red blood cell, intestinal mucosa, brain, and liver. Meaning to say that you don't need insulin for glucose to get into these tissues, to get inside. No? Meaning to traverse through your cell membrane. Now, glycolysis is occurring in the cytoplasm and it produces energy, the form of ATP, directly in the substrate level phosphorylation. It also produces NADH or a reducing equivalent, which is, of course, oxidized in the electron transport chain, meaning to say that NADH will have to go to your electron transport chain in order to be oxidized, and in the oxidation process, ATP are formed. Okay? It converts the glucose, your glycolysis, your glycolytic pathway, converts glucose into three carbon compounds. They are generally called triose phosphates, phosphorylated triose. And to be specific, they are glyceraldehyde 3-phosphate, and the other one is dihydroxyacetone phosphate. It is the glyceraldehyde 3-phosphate that continues to become or to finish the pathway, glycolytic pathway. The dihydroxyacetone phosphate will be converted to glyceraldehyde. So this will tell us that for every molecule of glucose, there will be 2 glyceraldehyde 3-phosphate that will be produced. And this goes on to another 5 reactions until pyruvate is formed. Okay? And it also says here, glucokinase is a lever enzyme for glucose. And this is, of course, very, very specific for glucose. And we will have a distinction between these two later on. Now, while the other enzyme, hexokinase, which does the same thing as glucokinase, is located in other cells other than the hepatic cells. And hexokinase is not so specific. It acts on other hexoses. Now, aside from glucose, it can act on fructose, mannose, galactose, other hexoses. All right. In the glycolytic pathway, there are 3 irreversible reactions, which is catalyzed by hexokinase or glucokinase. Now you know already if it is hexokinase, it is not liver cells. If it is glucokinase, it means a liver cell. Okay? So glucose is converted to glucose 6-phosphate. And then that glucose 6-phosphate is isomerized by an isomerase to become fructose 6-phosphate. And then another phosphorylation will occur. Addition of another phosphate to fructose 6-phosphate. And this is catalyzed by phosphofructokinase and produces fructose 1,6 biphosphate or 1,6 diphosphate. Okay? A series of reactions occur until you come up with phosphoenol pyruvate. Phosphoenol pyruvate is a high energy compound and be able to add a phosphate, a high energy phosphate to ADP to become ATP. And this is catalyzed by pyruvate kinase. So phosphoenol pyruvate becomes pyruvate. So 3 irreversible reactions at the level of glucose becoming glucose 6-phosphate, at the level of fructose 6-phosphate to become fructose 1,6 biphosphate, and at the level of phosphoenol pyruvate to become pyruvate. Okay? Right, let's continue. Let's talk about the regulatory enzymes. Those 3 irreversible steps are also regulatory enzymes. No? They are also regulatory enzymes. So the first irreversible reaction, it's either glucokinase or hexokinase. So let's talk about glucokinase first. It's a liver enzyme. You cannot find it in any other tissue except in the liver. Now, in the liver, there is no hexokinase. But these two enzymes, glucokinase and hexokinase, can do the same reaction. And we call them isozymes. You remember? Isozymes. Enzymes that catalyze the same reaction, but they differ structurally, chemically. Okay? All right. So glucokinase, a liver enzyme specific for glucose. It has a high Km, which means a low affinity for its substrate, and the substrate is glucose. It has a high Vmax. Catalyzes glucose phosphorylation, converting free glucose into glucose 6-phosphate. Insulin facilitates its action, and the reaction is, of course, irreversible. Now, this enzyme, glucokinase, is responsible to handle the increase of glucose after absorption in the intestinal tract. You know what's going to happen there in the intestinal tract? When you have already hexosis as a result of digestion, this, of course, is transferred from the walls of the intestine into the blood vessel which surrounds the intestine. And these are your veins that are around. No? And they are the ones that absorb or From absorbed, it goes directly to the blood. And these small veins, they coalesce and become a bigger vein, which we call the portal vein. And this portal vein goes directly into the liver. So that the liver is the first organ that receives all nutrients that has been absorbed in the intestine. So after eating, of course, and digesting the food that you have taken in, and if it has sugar or carbohydrates, it's turned into glucose. The glucose now is very high in concentration in the portal vein, which goes directly to the liver. And so, your glucokinase is suited for this because it has a high Km, meaning to say it requires a large amount of substrate before it reacts. And so it has a low affinity for the glucose. Okay, that's the important thing there. It is responsible for the increase of glucose concentration that goes into the blood. Goes into the liver. Now, hexokinase is found in every cell except the liver cell. It is less specific because it can act on mannose, fructose, yeah, of course, glucose and galactose. It can act. It is inhibited by its own product, glucose-6-phosphate, while glucokinase is not inhibited by its own end product. Okay? And in contrast to glucokinase, hexokinase has low Km. So which it only requires a low concentration of your glucose in the blood where it can now react. Okay, so how much is this? When we say high Km, low Km, high Km for glucokinase, high Km for glucose, that would require about 10 millimoles of glucose, 10 millimoles. While hexokinase is 0.1, 0.1 millimoles. So that's a big contrast from 10 to 0.1. So this tells you that a very, very low concentration, that's 0.1 millimoles already, your hexokinase still works. Okay? So that is 0.1, that is your hexokinase still even works at a maximum rate at fasting blood glucose levels. And the fasting blood glucose level is 5 millimoles and below. That's your fasting blood levels, glucose, 5 millimoles and below. Okay. So, but then that 5 millimoles is still high because it only requires 0.1 millimoles for hexokinase to work. So you can just say how efficient is hexokinase in phosphorylating glucose. All right, the next one is phosphofructokinase 1. This is a rate-limiting enzyme. The action is facilitated by a high concentration of AMP. Now, when you have a high concentration of AMP, it means that the cells is in a low charge, meaning ATP has been used because it has now become AMP. Okay, so that's why phosphofructokinase, a rate-limiting enzyme, would go into action when the concentration of AMP is high. And another, if the fructose 2,6 biphosphate is also high. No? So either of the two can activate your phosphofructokinase. High AMP and also high fructose 2,6 biphosphate. Now, how is it inhibited? It is inhibited by citrate. Okay, this is very interesting. Where do you find citrate? In what pathway? Krebs cycle. Other name of Krebs cycle is tricarboxylic acid cycle. Call it tricarboxylic because there are 3 carboxyl groups of citrate. And because of that, it is also known as citric acid cycle. No? So do not forget, no? Citrate. Allosterically inhibited by citrate. When you have plenty of citrate already, which means that you have been using glucose, converting it into pyruvate, and that pyruvate goes into the mitochondrion and converted to acetyl-CoA, and acetyl-CoA combines with oxaloacetate and forms citrate. So which means that the glycolytic pathway is working. No? So there's plenty of acetyl-CoA. Okay. And then another allosterical inhibitor for phosphofructokinase is a high concentration of ATP. No? High concentration of ATP. So which means there's plenty of ATP already, there's no need. So we have to inhibit, no, the phosphofructokinase. Okay. So the reaction is irreversible. All right. Then the last two, wait a minute. Remember that again, fructose 2,6 biphosphate. Fructose 2,6 biphosphate is formed from fructose 6-phosphate by phosphofructokinase 2. Now, we were talking of phosphofructokinase 1. Now, 2,6 biphosphate is catalyzed by phosphofructokinase 2. Okay? Phosphofructokinase 2. So after a meal, fructose 2,6 biphosphate is high. Okay? It's high. So phosphofructokinase 1 is activated. and glycolysis is stimulated. Because remember, phosphofructokinase 1 is the rate-limiting enzyme of your glycolytic pathway. You get it? Yeah. Now, where is this phosphofructokinase 2? This is found in the liver. Phosphofructokinase 1 is all over tissues, including the liver. Okay? All right. The next one is pyruvate kinase. It converts phosphoenol pyruvate, a high energy compound, to pyruvate after phosphorylating ADP to ATP by substrate level phosphorylation. Okay. Pyruvate kinase is inhibited in the liver. Okay, it's only in the liver. Pyruvate kinase is inhibited in the liver during fasting, but it is not inhibited in other tissues. So, during fasting, when glucagon levels are high, glucagon, we have not taken up hormones, but we'll just tell you that glucagon is a hunger hormone. You feel hungry. When you feel hungry, glucagon is secreted. Now, glucagon is opposite to insulin. While insulin is produced in the pancreas, in the islets of Langerhans, produced by the beta cells of the islets of Langerhans, your glucagon is produced in the same islet but different cells, alpha cells of the islets. So these two are antagonistic, or I mean not really antagonistic, but they have opposite action. They have opposite action. When there is so much glucose in the blood, insulin is secreted. When there is no more glucose in the blood, or there is very low glucose in the blood, glucagon is secreted. You get that? Yeah. So glucagon is secreted via hunger signal is cyclic AMP. Cyclic AMP activates a protein kinase. Okay, a protein kinase which is going to phosphorylate. When it's a kinase, it phosphorylates. Okay. When pyruvate kinase is inhibited, you are going to promote gluconeogenesis. What is gluconeogenesis? It's the conversion of non-carbohydrate sources into glucose. We will be talking about that now as we go along. Okay. So you have now the summary. Glycolysis is the cytosolic pathway in all mammalian cells for the metabolism of glucose or glycogen to pyruvate and lactate. It can function anaerobically by regenerating oxidized NAD required in the glyceraldehyde 3-phosphate dehydrogenase reaction by reducing pyruvate to lactate. Lactate is the end product of glycolysis under anaerobic condition. Example is in exercising muscle and in erythrocytes. Okay, and in erythrocytes. The glycolysis in red blood cell is anaerobic. It is very paradoxical because RBC carries oxygen, and yet it cannot use the oxygen that it carries. So the glycolytic pathway in RBC is anaerobic. And that is the reason why when there is low concentration of glucose, the glucose is always conserved for the use of RBC because other tissues can go to the utilization of other sources of fuel, no? Because they can use any other fuel. RBC cannot use any other fuel, only glucose, only glucose. Now the reason why it cannot use the oxygen that it carries, your RBC cannot use the oxygen it carries, it is because it does not have the apparatus anymore for the utilization of oxygen. The utilization of oxygen requires what you call your respiratory chain. You still remember respiratory chain or electron transport chain? At the end, there is the union of hydrogen and oxygen to form water. No? Now, it does not have mitochondrion. Now, you know that your RBC does not have any organelle anymore. Histologically, you know that immature RBC contains organelles. But when they mature, the organelles are removed and replaced by hemoglobin molecules. 240 or more. Hemoglobin molecules. And that is just one that your RBC carries. Okay. All right, lactate and acid is an aerobic condition and exercising muscle. Okay, why do the muscles undergo anaerobic glycolysis also? First, it is aerobic, exercising muscle. I mean, when you exercise, or a very good example of this is in a marathon exercise. Like for example, a marathon running, marathon run, and marathon swimming. Why? We know that your muscles uses oxygen, okay? But when it is in exercise, usually in prolonged exercise, like in marathons, the delivery of oxygen to the muscles could not cope up with the demand of oxygen. by the mitochondrion. Now remember that your blood carries that oxygen into the muscles and load the oxygen, and it goes back to the lungs. And that travel going back to the lungs, and in the lungs it is aerated, no? Oxygen is carried, and it is not just instant, it takes a little while. So that in the long run, in the long exercise, the supply of oxygen becomes deficient because the delivery could not cope up with the demand of the muscle. So that's why the muscle goes into an aerobic condition. And so what happens? We know that in anaerobic glycolysis, lactic acid is produced. Now we know that lactic acid is a weak acid, but if there's so much of lactic acid, it will change the pH of the environment. And naturally, the pH there of the cell goes low. And the enzymes cannot work because they are usually exposed only to the physiologic pH, which is 7.4. And now if that goes down because of lactic acid accumulation, so the cells, especially the muscle cells, will not work anymore. And it will give you cramps. So when it cramps, what happens? You have to stop. Okay? You have to stop. So the muscle tells you, you have to stop because we are already anaerobic. We do not have oxygen anymore. But you are still hard-headed. You keep on. So everything now stops. And the muscle just contracts without relaxing. You cannot go on anymore. You have to stop. Now when you stop, in a little while, the delivery of oxygen now is restored. So the muscle cells will now revert to aerobic conditions. Okay? There is a reversal now of lactic acid becoming pyruvate. And pyruvate goes into the mitochondrion to become acetyl-CoA. Acetyl-CoA used by citric acid cycle, you will have now plenty of energy in the form of ATP. Did you get that? Okay. So glycolysis is regulated by 3 enzymes catalyzing non-equilibrium reactions: hexokinase, phosphofructokinase, and pyruvate kinase. In erythrocytes, the first site in glycolysis for generation of ATP may be bypassed for the formation of 2,3-biphosphoglyseric acid, which is important in decreasing the affinity of hemoglobin for oxygen. Pyruvate is oxidized to acetyl-CoA by a multi-enzyme complex known as the pyruvate dehydrogenase, which is dependent on vitamin-derived cofactor thiamine diphosphate. Conditions that involve an impairment of pyruvate metabolism frequently lead to lactic acidosis. Lactic acidosis. Okay. Let's go now. Let's go now to the next chapter. Before that, let's talk about your questions first. Your questions. I have like to ask you if you have questions. So we can clear this up. You have questions? Wala. Hello? Anybody there?
We're here po, Doc.
Hello, Doc.
Okay. Do you have questions?